So thank you to Carol and the organisers for the opportunity to participate in this session on targeting nuclear steroid receptors. Resistance to standard of care ER target therapies is the main cause of breast cancer mortality, necessitating new strategies for treating or ideally preventing resistance. I've spent most of my career studying the androgen receptor in prostate and breast cancer. Today I'll present some of our latest work, providing what I believe is a paradigm-changing new perspective on how to treat ER-positive breast cancer by adopting AR agonism as an alternative form of endocrine therapy. Before proceeding, I declare I have nothing to disclose. The androgen receptor is expressed in almost all breast cancers, with highest expression in ER-positive tumours, making it a prevalent therapeutic target. Most recently, we showed that the androgen receptor are co-expressed in about 60 to 70% of tumour cells in DCIS, invasive ductal carcinoma, and lymph node metastases, as illustrated by the immunostaining for an invasive ductal carcinoma shown in this panel, where the estrogen receptor is labelled in red, the androgen receptor in green, with the majority of nuclei shown in this inset being orange, indicating co-localization of the estrogen receptor and androgen receptor. This is consistent with the potential for a direct interaction between these two signaling pathways. Androgen receptor expression is associated with low proliferative index and small tumour size, and importantly, the androgen receptor is an independent predictor of overall survival in ER-positive disease. Historically, androgenic drugs were used as a therapy for advanced breast cancer, with evidence of clinical efficacy, as reported by Kennedy in the New England Journal of Medicine in 1958. Interest in androgenic therapy for metastatic breast cancer continued into the early 1990s. For example, Ingle and colleagues at the Mayo Clinic demonstrated an additive benefit of tamoxifen and fluoxymesterone in a subset of patients, even in the absence of a biomarker for patient selection. However, how best to target the androgen receptor in ER-positive breast cancer has been a major clinical controversy over the past decade. Because the androgen receptor is oncogenic in prostate cancer and AR antagonists are an effective treatment for that disease, it was generally assumed that if the androgen receptor is expressed in breast cancer, then an AR antagonist would be the appropriate treatment strategy. This view was fueled by the desire to repurpose anti-androgenic drugs used to treat prostate cancer and by equivocal preclinical breast cancer studies. Moreover, in the absence of definitive preclinical data, AR antagonists and agonists strategies have been and still are being trialled concurrently in the same ER-positive breast cancer populations. This highlighted to us the importance of generating robust in vivo data with contemporary preclinical models to resolve this clinical controversy. Initially, we interrogated human breast tissues using an ex vivo patient-derived explant or PDE model, where human breast tissue fragments are cultured on a gelatin sponge to sustain tissue complexity and sex hormone responsiveness. Antagonistic sex hormone activity was evident in normal and malignant breast epithelial cells when assessing incorporation of BRDU, a marker of newly synthesized DNA. Estradiol or E2 stimulated BRDU incorporation into both normal breast and malignant breast tissues, and this effect of E2 was abrogated by co-treatment with dihydrotestosterone, DHT, the most potent endogenous androgen receptor agonist. Quantification of Ki67 immunostaining in the PDEs demonstrated that co-treatment with E2 plus DHT significantly reduced the proliferative index compared to E2 treatment alone. Consistent with these findings, E2 positively and E2 plus DHT negatively correlated with gene signatures of cell cycle regulation based on our RNA-seq data. Collectively, these findings indicate that AR agonism is anti-proliferative in both the normal breast and primary ER-positive tumours. We further evaluated the efficacy of AR agonism in vivo using a ZR75-1 xenograft model, selected in part because it has robust expression of the androgen receptor and the estrogen receptor, and tumour growth is stimulated by estradiol. The key feature to note from this slide is the durable inhibition of E2-induced growth by both dihydrotestosterone in green and the selective androgen receptor modulator enobosarm in red. Dual immunostaining 5 days after initiation of treatment highlighted how Ki67 in blue in the E2 treated tumours is markedly suppressed by co-treatment with enobosarm, which also, as can be seen from this right-hand panel, markedly increased androgen receptor levels shown in red. To confirm our preclinical findings using the ZR75-1 xenograft model, we undertook studies using contemporary patient-derived xenograft or PDX models of ER-positive breast cancer derived from patients with treatment-resistant metastatic disease. Estradiol stimulated tumour growth in the UCD4 PDX model, which expresses a clinically relevant ESR1 gene mutation. But E2-induced growth was suppressed by combination treatment with either a SARM or dihydrotestosterone. We replicated these findings in other estrogen-sensitive PDX lines, including the HCI-005 model, originally developed by Elena Welm at the University of Utah. This model also expresses a mutant ESR1. Again, E2, shown in orange, stimulated tumour growth, and this was potently and durably inhibited by co-treatment with dihydrotestosterone, shown in blue. Notably, the AR antagonist, enzalutamide, shown in red, had no effect on estrogen-stimulated tumour growth. But enzalutamide partially reversed the growth inhibitory effect of dihydrotestosterone, as expected for an anti-androgen, suggesting that treatment with enzalutamide could potentially inhibit the beneficial effect of endogenous AR activity in a breast tumour. To address the mechanism of AR agonist inhibition of ER-driven tumour growth, we first performed ChIP-seq and RNA-seq experiments using the ZR75-1 breast cancer cell line. The AR and ER ChIP-seq data is represented in this two-factor log ratio M plot. The left-hand quadrants show ER binding sites that are lost upon AR activation with dihydrotestosterone, including loci associated with established ER target genes such as the progesterone receptor and MIB. The upper right-hand quadrant shows ER and AR binding sites that are gained upon AR activation. These gained ER and AR binding sites include loci associated with known AR regulated genes such as SEC14L2 and EAF2. This is illustrated by representative genome browser images. The middle panel here shows androgen-induced loss of ER binding at ER target genes. The two examples show loss of ER binding at loci within intron 1 of the MIB gene and the enhancer region of the progesterone receptor gene. And this corresponds to a downregulation of MIB and PGR expression in the RNA-seq data. Conversely, AR agonism increased both AR and ER at AR target genes such as EAF2 in the top upper panel and SEC14L2 in the bottom panel. And again, this was associated with a clear increase in EAF2 and SEC14L2 expression in the RNA-seq data. These genome browser examples of ER and AR binding events are illustrative of what occurs at all ER target genes downregulated by DHT and all AR target genes that are upregulated by DHT. Importantly, we have confirmed these findings using in vivo ChIP-seq to analyse tumours from AR agonist-treated PDXs. In addition to repositioning AR activation repositioned the critical ER co-activators SRC3 and P300 from ER to AR target genes. As shown in the two-factor log ratio M plot, AR activation caused a major redistribution of E2-stimulated P300 and SRC3 binding sites, with binding sites being lost at ER binding sites, including the progesterone receptor and BCL2 and MIB, and gained at AR binding sites, including EAF2 and SEC14L2. Consistent with this relocation from ER to AR binding sites, DNA motif analysis revealed that loss of P300 SRC3 occupancy occurred at estrogen response elements, and gain of P300 SRC3 occupancy occurred at androgen response elements. Hence, sequestration of key transcriptional cofactors plays a key role in estrogen receptor and androgen receptor crosstalk in breast cancer cells. I've summarised our current understanding of the mechanism of AR agonist-induced growth inhibition of ER-positive breast cancer in this cartoon. Activation of AR displaces ER binding and critical co-activators, including P300 and SRC3 from chromatin at ER-regulated genes such as NIC, MIB, and cyclin D1, leading to their transcriptional downregulation. In turn, sequestration of P300 and SRC3 by AR facilitates upregulation of canonical AR target genes, including known tumour suppressor genes. Collectively, these two mechanisms, that is, loss of ER and activation of AR-regulated genes, results in suppression of ER-driven breast cancer growth. The detailed mechanism is described in our Nature Medicine paper published earlier this year, which was driven by Teresa Hickey from our group in Adelaide and undertaken in collaboration with Jason Carroll at Cancer Research UK's Cambridge Institute and Jean Lim at the Garvan Institute in Sydney. If you're interested, we made a cartoon video explaining the key aspects of these findings. We next asked, is AR agonism additive in combination with standard of care ER target therapy such as tamoxifen? In the HCI-005 PDX model, which is estrogen sensitive, DHT, shown in blue, more effectively inhibited tumour growth than the ER antagonist tamoxifen, shown here in red. Importantly, the combination of DHT plus tamoxifen in black was similar to treatment with DHT alone. Immunohistochemical analysis of tumours at 7 days of treatment, shown here, revealed that DHT markedly increased AR nuclear protein levels and suppressed the proliferation marker Ki67, consistent with its effect on tumour growth. The same was observed for the combination of DHT plus tamoxifen on both AR protein levels and Ki67. We were particularly interested, given this finding, in whether AR agonism also inhibits growth of an estrogen-independent ER-positive model that is more reflective of resistance to aromatase inhibitors, as treatment resistance in this disease setting is a major cause of breast cancer deaths. In collaboration with Jean Lim at the Garvan Institute in Sydney, we developed the endocrine-resistant GAR1513 PDX model, which was derived from a liver metastasis of a patient resistant to aromatase inhibitors and fulvestrant. The GAR1513 model is strongly ER positive, but does not require exogenous estradiol for robust basal growth in vivo. Treatment with DHT significantly reduced the growth of GAR1513 tumours, whereas treatment with the AR antagonist enzalutamide had no effect on tumour growth. These findings indicate that AR agonism can inhibit ligand-independent ER activity that drives many endocrine-resistant tumours. Given this finding, we investigated the efficacy of a cyclin-dependent kinase 4/6 inhibitor in the E2-independent GAR1513 model, alone in combination with an AR agonist. Tumour growth in the GAR1513 PDX model is partially inhibited by the CDK4/6 inhibitor palbociclib, shown in green, but not to the same extent as with the AR agonist enobosarm, shown in purple. Importantly, the combination of the AR agonist and palbociclib had a superior growth inhibitory effect compared to either agent alone. While this finding is very encouraging, we were particularly interested in whether AR agonism is growth inhibitory in a model resistant to a CDK4/6 inhibitor, especially as the development of resistance to CDK4/6 inhibitors is considered inevitable. Hence, we investigated the combination of an AR agonist enobosarm with palbociclib in a newly developed PDX model, which originated from a liver metastasis of a patient who relapsed on aromatase inhibitors tamoxifen, fulvestrant, and palbociclib. As expected, this model was resistant to palbociclib, shown in red. Activation of the AR was tumour suppressive, but most importantly, the combination of the SARM plus palbociclib had an additive effect. The proliferative index, assessed by Ki67 immunohistochemistry in tumours at 7 days post-treatment, was markedly reduced in the combination arm, as shown in the bottom right-hand panel, consistent with the tumour growth at endpoint. Quantification of the Ki67 is shown in the right-hand side panel. Details of this preclinical study can be found in the poster presented in a spotlight session earlier today by Allegra Freelander a PhD student at the Garvan Institute in Sydney. In conclusion, our preclinical studies clearly demonstrate that AR has a tumour suppressor role in ER-positive breast cancer, and that AR agonists, either alone or in combination with standard of care ER target therapies or CDK4/6 inhibitors, confers potent and durable growth inhibition, indicating that AR agonism is a viable strategy to inhibit the growth of ER-positive breast cancer. Importantly, our findings have informed the design of new clinical trials with selective androgen receptor modulators, which will establish whether AR agonism is a new paradigm of endocrine therapy for ER-positive breast cancer. Finally, while I've acknowledged the key people who have contributed to this study throughout the talk, such as Teresa Hickey and Jason Carroll and Elgene Lim, I would also like to acknowledge the many other people who made critical contributions to this study as shown. I'd also like to acknowledge our key funders, the National Breast Cancer Foundation and the National Health and Medical Research Council of Australia, and a special thanks to those many men and women who supported our research. So thank you for your attention, and I'd be delighted to take any questions.